Yeah, hi and good afternoon to today's edition of MycoTalks. This is the last edition before our summer break, and it's a great pleasure for me to chair this together with Joe Heitman. Before we start with our invited talks, I would like to say a few brief words about the whole purpose of this seminar series. Today we will have two speakers, as always. The first one is going to be Darius Armstrong-James from Imperial College, and the second one, Aaron Mitchell from University of Georgia. I'm chairing today, as I said, together with Joe Heitman, on behalf of the organizing committee of the MycoTalks series, which is shown on this slide. The whole series is organized by the MRC Center for Medical Mycology at the University of Exeter, and there are numerous activities, including a Twitter account that you can follow. The idea of the series is to really present outstanding research and advancements in the whole field of fungal infections, fungal disease, fungal pathogens. Besides quality, the seminar series also tries to provide the whole range of career stages, gender, geography in the field. So diversity is a very important aspect. We will have two 30-minute talks by the two speakers. And afterwards, 30 minutes of in-depth discussion with both speakers. It's very important for that you submit your questions through the Q&A function. Ideally, you start with the name of the speaker so that Joe and I can sort the questions to the speaker. And we will then, in the end, ask all the questions. But it doesn't matter at all if the questions keep popping in during the talks. As always, the talks will be recorded and the recordings will be made available at the YouTube channel. And I've heard that this is now eagerly used. Yeah, we will have a summer break in August, so no MycoTalks in August, but there is a reason to look forward to autumn and September, and we start again on the 28th of September with two talks from Jay Vyas and Carol Monroe. Before we go into today's talk, I would also like to point your attention towards the trainee speaker series, the Medical Mycology Trainee Seminar Series. So this is directed towards the rising stars in the field, can be students, postdocs, clinical trainees, and also new faculty. And I think this is really an outstanding chance to present your work to the global community, something that is very well taken up and visible. And this is always on the second Thursday of the month. And we really invite people to get in touch or submit abstracts if you feel that this is your chance to present your work. Now, with this, we will go right into the topics today. And our first speaker is Darius Armstrong-James. And it's a real pleasure, Darius, to welcome you here. Darius is a professor in infectious diseases and medical mycology at Imperial College in London. He's also a consultant with several other hospitals. I guess that's really cool because this expertise is not that frequent, and it's good to share it with different hospitals. But Darius, as many of you will know, has also continuously done cutting-edge research, and I guess this goes back to his former times where he has been in the labs of the very big names in the field, and I read Ken Haynes in the CV and also John Perfect. But today, of course, he is a big name by himself, and therefore it's really cool to have you here, Darius. That your focus has been Aspergillus and Aspergillosis, but I guess you've also been working with other fungi. And today, as I get from the title, it's about Aspergillosis and cystic fibrosis, and I'm really looking forward to this.
For the kind invite as well. It's great to be here and talk about our work. So I'm just going to share screen. Can you see the slides?
Yep, perfect.
Great. Okay, I'll crack on then. Thank you. So yeah, today I'm going to talk about some of the work that we've been doing that's really been driven very much by clinical isolates in essence. So phenotypic and clinical correlates of genome diversity in Aspergillus fumigatus. So I'm just going to firstly talk a little bit around clinical data underpinning immunophenotype switching in chronic respiratory aspergillosis, which is very much part of our clinical practice in London. Then talk about some of the phenotypic correlates that we see for genomic diversity in Aspergillus fumigatus. And then finally finish up with trying to bring through some of that work, correlating these clinical phenotypes in clinically relevant animal models. So as the audience will know, that we're exposed to fungi on a daily basis, and amongst these, really, the Ascomycetes and the Basidiomycetes are the two groups that are really causing severe fungal disease. I'm particularly interested in Aspergillus, which is an Ascomycete, which is ubiquitous in the environment and continuously inhaled by everyone on the planet. It reproduces by asexual reproduction primarily, although sexual reproduction and parasexual reproduction also occur, and has these very small 3-micron spores generally, as you can see here, which are basically inhaled right down into the small airways of the lung. And in my lab, historically, we've been quite interested in the interface with macrophages. So here on the right, you can see green Aspergillus spores and macrophages in culture. And if you just follow this macrophage here, what we're really interested in is the host pathogen interface and whether or not that macrophage can control the fungus or whether it is able to germinate. And when it germinates, of course, it basically transitions into a multicellular hyphal phase. And in this particular instance here, it's actually killed that macrophage. So if an infection does ensue during those events in the lung, then there's a number of different outcomes that may occur. And amongst these are invasive aspergillosis and chronic pulmonary aspergillosis in the context of weakened immunity, colonization, which occurs primarily in patients with chronic respiratory diseases, and then in people who have allergic sensitization, they may also develop either severe asthma with fungal sensitization or allergic bronchopulmonary aspergillosis, which is the most severe form of fungal sensitization that we see in the lungs. So there's a remarkable spectrum of aspergillosis, and I think this probably mimics many fungal disease, that you can get allergic outcomes as well as inflammatory outcomes depending on the host immune status and the behavior of the organism. With invasive aspergillosis, of course, we see these macronodules in the lung, which are really just mycelial balls, which may be, a single organism essentially. And around those, you can see this ground glass appearance, which is in essence hyphal invasion, neutrophilic inflammation, and also a particular feature is angioinvasion into the lung blood vessels, where you can see subsequent hemorrhage. And of course, this autopsy specimen here, you can see abundant hemorrhage around this Aspergillus mycetoma in this patient's lung. There are two forms of invasive aspergillosis, primarily, so neutropenic, where you see minimal inflammation and there's abundant hyphal invasion, and then the non-neutrophilic, neutropenic context, which is primarily driven by steroids or other immunosuppressants. And in that context, you actually tend to see inflammatory necrosis. These are actually clinical histopathology examples here. In chronic aspergillosis, this behaves very much like tuberculosis. So here you can see this mycetoma within a cavity in the lung, which was basically generated by previous tuberculosis. And the interface between TB and Aspergillus is a specific interest in the lung, which I won't talk about today. Allergic bronchopulmonary aspergillosis is really just at the other end of the spectrum. And this disease, essentially, within the context of asthma or cystic fibrosis, Aspergillus is able to propagate within the airway. And here you can see this sausage-like protuberance is essentially called the hyperdense mucus sign, which is where there is abundant hyphal growth within the airway, leading to dilatation of the airway and subsequently bronchiectasis and surrounding inflammation. And this inflammation, as you can see in this schematic here, is really driven by Th2, Th17 responses and dendritic cell T cell interface in the context of asthmatic atopy. So what's actually quite interesting about this spectrum of disease is that actually you can generate this and recapitulate it in the mouse model. So here, this is work from David Corrie's group, and what you can see is that at low doses of Aspergillus on a repeat intranasal challenge, you basically generate allergic inflammation characterized by eosinophilic infiltration and IL-4 release. But if you up the dose, then you can switch this and basically flip to neutrophilic inflammation and completely abrogate the eosinophilic component, and that's driven by interferon gamma. So this really phenocopies what we see in our human patients. So basically speaks slightly to the issue of B-cell isotype switching and antigenic load. This is something that we also see in cystic fibrosis. So this is now clinical data from Manchester, where they looked at the spectrum of disease in cystic fibrosis and were able to classify patients using latent class analysis into 4 different classes, class 1, 2, 3, and 4, which are basically colonization, ABPA, sensitization, and Aspergillus bronchitis, which is an inflammatory airway disease. And what's notable there is that this all seems to relate to IgE and IgG responses. The allergic patients have IgE responses, and the bronchitic patients have IgG responses. And interestingly, those bronchitic responses seem to correlate very well with Aspergillus IgG, sputum galactomannan, and also sputum Aspergillus PCR. And what this suggests is that it's actually the fungal burden, very similar to the animal models, that's defining whether or not there's an allergic phenotype with a low fungal burden or an inflammatory infection phenotype with a high fungal burden. So that seems to be a reproducible relationship, both in the murine and clinical setting. So what we actually see clinically is that with this sort of damage response framework, that you have this kind of interface in CF where patients are switching between bronchitis and sensitization or colonization. But in addition, they may develop either ABPA or chronic pulmonary aspergillosis. And you can actually get immunophenotype switching between those 2 conditions. And this seems to relate to how they are treated and managed. So if a patient has chronic pulmonary aspergillosis and you put them on treatment, they can basically develop what we think is an overlap syndrome, where they develop sensitization characterized by eosinophilic inflammation and high IgE. And on the other hand, if you have a patient with allergic bronchopulmonary aspergillosis and you give them steroids or an eosinophil blocking drug like mepolizumab, they can actually go on to develop chronic pulmonary aspergillosis and even invasive aspergillosis. So there's actually a whole cycle here of immunophenotype switching that we see clinically. So to look at this in a bit more detail, we conducted a retrospective study at the Brompton in around 400 patients that we treated with ABPA, which is obviously quite a large group, and I think speaks to the fact that the UK is one of the big centres, along with probably India, for ABPA. So this is over a 10-year period, primarily white and Asian patients. And we started looking at these very useful serological markers we have to look at their immunophenotype. And in essence, what we saw is that those patients who were developing bronchiectatic ABPA, i.e. Aspergillus causing airway destruction, there was a higher Aspergillus IgG consistent with increased fungal burden in the lung. And this was associated with a reduced FEV1. So the bronchiectasis that we see in these patients doesn't seem to be associated with the IgE phenomenon, the sensitization phenomenon, but more with the IgG phenomenon and the airway fungal load, in essence. And when we looked at the impact of IgG on lung function, we saw that the IgG had a negative impact on lung function in terms of FEV1. And if we treated patients with ABPA with antifungals, then we could basically reverse the IgG response and improve the FEV1. And this has been a contentious area in ABPA, this question of whether or not antifungals are actually appropriate in this setting or whether steroids are the right way to go. So when we compared this to steroids, what we actually saw was that in terms of FEV1, steroids over time are actually fairly ineffective, whereas the antifungals itraconazole and posaconazole seem to drive improvements in FEV1 over years, actually. So it's quite difficult to generate this kind of data, but we've been able to do it using data informatics in our center. And posaconazole, which is a highly active antifungal, basically reduces the IgG over time. But when you look at steroids, they actually increase the IgG, which is not surprising because they're impairing the immune system. So steroids in the short term are very good for ABPA in terms of reducing down mucus production and inflammation. But in the longer term, it looks like they're actually impairing fungal immunity, leading to declining lung function, presumably related to increasing airway-based inflammation. So in summary, there's a spectrum of disease in chronic respiratory aspergillosis from CPA to ABPA, and we see immunophenotype switching in cystic fibrosis from ABPA to bronchitis, and increased fungal load in ABPA, which seems to drive switching from IgE to IgG responses under steroid pressure. And this actually speaks to this broader concept of hypersensitivity reactions in the immune system. And with our sort of simple IgE-driven disease in ABPA, what we actually see is a type 1 type response, which is driven by IgE, leading to mast cell degranulation and eosinophilic infiltration. However, in these more complex patients with increasing fungal burden, it looks like they're flipping to a type 3, type 4 response, where we're seeing IgG-based humoral responses leading to activation of granulocytes and increasing airway inflammation. And so just summing up, the increased fungal load that we see drives IgG responses, and this is exacerbated by steroids and other immune suppressants, and the antifungals drive it the other way. They seem to be able to flip people into allergic responses. We see this quite often in immunosuppressed patients with aspergillosis. So just moving gears slightly, I'm now going to flip on to talking about clinical isolates. And I'm just going to frame this by discussing the aspects of triazole resistance in Aspergillus, because this is really how we got into the whole clinical isolate game. And of course, many of you will know that actually it was David Denning back in the US in the 90s who first described itraconazole resistance. But in fact, the big studies from the Netherlands and from Manchester came out in the noughties, essentially showing this incremental resistance in Aspergillus to triazoles. And within that context, it's actually been epidemiologically interesting to study this, because we know that there are mutations that occur due to treatment within host, such as G54 and M2220, which occur in the target of the triazole antifungal CYP51A or ERG11, whereas there are agricultural drivers for resistance due to fungicide use, which lead to tannin repeats in the promoter region of CYP51 and typical mutations such as L98H. So you can basically tell in your patient whether they've acquired resistance from the environment or from in vitro treatment of that patient in situ. And we've been studying this in detail for a Wellcome Trust Collaborative Award that was led by Matt Fisher with collaborators from Manchester, Exeter, Nottingham Kings, and Radboud in the Netherlands, and we've done large-scale population genomics looking at the emergence of environmental resistance and how that's impacting on our clinical cohorts. And we essentially showed that there are two clades of Aspergillus shown here, and within those clades, most of the environmental resistance is in clade A, which is the agricultural clade. And within that, there seems to be this clade AA, which is spreading clonally in northwestern Europe, associated with environmental resistance. And when we studied our patients at the Brompton, what we actually found is that within this clade AA, which you can see here, there are essentially clonal isolates which are shared between the environment and our patients that have environmental resistance mutations. And so that's kind of strong molecular evidence that these environmental resistance isolates are getting into our patients. So following on from that, we decided that we would study the clinical isolates from our patients with chronic respiratory fungal disease in further detail. And this is basically a similar phylogenetic map of clinical isolates here, showing again that they still have the same population structure with clade A and clade B. And in essence, these are mostly from cystic fibrosis patients. 50% of them are triazole resistant, not because that's our rate of resistance, but more because we were enriching for resistant isolates, and there are many different mutations, which you can see here in the panel. We decided that we would submit these isolates to phenotypic microarray culturomics using the Omnilog platform, which we acquired through the NIHR funding stream. And in essence, this allows us to test multiple conditions in parallel using precast plates, essentially, with different substrates on them, focusing on carbon and nitrogen source, osmosis stress, and pH stress. And just having an eagle's eye view of the response of the isolates, you've got the isolates on the y-axis and the conditions on the right, on the x-axis. It's pretty clear that Aspergillus have some conditions where it grows very well, and that there's significant phenotypic diversity in terms of how these organisms behave at a population level. When we looked simply at the metabolic set enrichment analysis to look at what the main substrates for Aspergillus, the main things that came out across the population were metabolism of extracellular glycans, amino acids, diverse lipids, and oxidation-related processes. And we then started to compare isolates from different patient groups. So here we're looking at CF and non-CF patients, so cystic fibrosis. And when you look at metabolism of carbon and nitrogen, what you actually see on the Omnilog is there are 13 metabolites that can actually discriminate CF from non-CF when you perform principal component analysis. And when we looked at what these metabolites are, the most striking one was actually L-histidine. So here you can see the specific comparison for L-histidine between patients with cystic fibrosis in blue and non-cystic fibrosis patients here. We then went on to look at osmolyte and pH flux, and it was pretty clear that the CF isolate seemed to be more able to grow in the presence of, in particular, chloride, which is, of course, the channel which is defective in CF. And so in a way, this is a nice positive internal control. These isolates do seem to have either adaptation or selection to the CF lung environment. And here you can see some of those comparisons for different salts between CF in red and non-CF isolates in blue. When we combined pH changes with metabolic flux, we saw even better separation of these isolates by principal component analysis. So in fact, it looks like pH-dependent control of different metabolites is particularly selected for by CF isolates that are preferentially growing in those conditions. We then looked at the resistant isolates versus sensitive isolates. And for this comparison, what came out really strongly was that sodium lactate seemed to be something that these resistant isolates seem to be metabolizing very well. And that's quite interesting because there's been quite a lot of work around hypoxia and aspergillosis, in particular from Rob Kramer. And we know that the lung is a hypoxic environment in these chronic patients. So again, some interesting biologically plausible observations here. And when we did pathway analysis, then it became clear that these resistant isolates had enhanced glycolipid and ketone metabolism. So these Aspergillus clinical isolates also divide into two clades, and using phenotypic microarrays, we can identify core metabolic components associated with disease endotype, with the example of salt tolerance in cystic fibrosis, histidine and histamine metabolism in ABPA, and lactate metabolism in triazole resistance. Of course, we need to confirm this at scale, but these are interesting preliminary findings. So moving on into immunology, we've basically been running a strategic research center looking at the immune response in cystic fibrosis to Aspergillus. And this is essentially primarily a clinical immunophenotyping exercise, which we haven't completed yet. However, we've been doing a fair amount of animal work through this strategic research center, essentially exploiting the repeat challenge model of David Corrie using live isolates, which I discussed at the start of this talk. And you can see that in this model, the mice do not lose weight with a repeat challenge of Aspergillus over 14 days at low dose, but they do develop mucosal inflammation, as you can see here, with mucin deposition, which you can see on the PAS stain there. And when we look at these mice, they, similar to the previous paper, they develop florid eosinophilic infiltration over a 14-day period associated with a loss of neutrophils, and interestingly also a loss of alveolar macrophages, and this is associated with a quite significant T cell and B cell proliferative response. And in addition, we see increased lactate dehydrogenase indicative of airway damage, which is useful within the context of bronchiectasis. And we also see that whilst there is not an increase in TNF, we do see that a number of these different inflammatory pathways that have been implicated in asthma, such as R1B, galectin-1, and R33 are all increased. So damage response pathways seem to be induced in this low dose repeat challenge model. We then undertook a proteomic analysis of these mice and identified a number of different proteins that were enriched within the airway. Amongst these, histidine-rich glycoprotein was of interest because of the histidine-related phenotype we'd seen in our clinical isolates. And when we basically did pathway analysis for the proteomic data, it was the R1 family complement pulmonary surfactants, chitinases, and calprotectin that came out as kind of dominant biological modules. And of course, those are all consistent with what we know about inflammation in the context of aspergillosis and allergy. And at the moment, we are looking at the R1 pathway, and in particular, when we inhibit the R1 receptor with wedanolactone, then we see that we can abrogate this R1 beta and R33 response, suggesting that caspase 11 is probably important for this. So I'll just finish by acknowledging the people involved in this work. So on the genomics and culturomics side, Joe Rhodes, Renad Al-Jahani, Matt Fish, Alison Holmes, and Gerard Leroy Mamous. And then with the models, Tom Williams, Xinshin Xu, Amelia Birkinson, Adelia Warris, our collaborator in Exeter, and Anand Shah. Thank you very much.
Yeah, thank you very much, Darius. Really cool. And I guess there must be some questions from the audience, so please remember to drop your questions via the Q&A function. And with this, I will directly hand over to Joe. You're still mute.
Thanks very much, Oliver and Darius. It's really a great pleasure to introduce Aaron Mitchell as our second MycoTalks speaker today. And Aaron is currently professor and head of the Department of Microbiology at the University of Georgia in Athens, Georgia. Now, Aaron is an icon in our field, and his brilliant science with model and pathogenic yeast has illuminated understanding the genetic control of meiosis, how pH is sensed, and how biofilms are constructed and influence biology and pathogenesis. We further owe Aaron a deep debt of gratitude for his vision in co-founding the Woods Hole Molecular Mycology course taught at the MBL each summer. And Aaron's dedication to medical mycology and medical mycologists has really forged our community and uplifted all of our science. And please welcome Aaron. We're very much looking forward to your talk, and thank you for joining us from Cape Cod.
Thanks very much, Joe. I will try to share my screen. I will try to fix my pointer.
Brilliant.
Great. And now I will try to give my talk. So thank you very much, Joe, for that very kind introduction. Today, I'm going to talk about some of our work on natural variation in Candida albicans. And specifically, we've focused, the studies I'll speak about today, we've focused on biofilm formation. So here's a side view of Candida albicans biofilm grown for 24 hours. You can see it's 1/4 millimeter thick, which is a lot thicker than you'd want inside of you. And the hyphae are readily apparent in the image, and it's a little harder to see in this particular kind of view, but there are plenty of yeast cells there as well. And one thing that, that we all agree upon in the field is that hyphae are required for biofilm formation. There's been a lot of different ways to look at that correlation, and they seem to hold quite well. Now, the reason that we're interested in biofilm formation is that biofilms are the basis for device-associated infection. So an implanted medical device, if it gets populated, if its surface gets populated by any microbe, that serves as a source of continuing infection. So fortunately, I can report that we've learned a lot about biofilm formation in Candida albicans. We know a lot of the gene products that are required to make a biofilm. We know the sort of sequence of events that occurs as biofilms form. We know many of the molecular components that are found in biofilms. And we know the regulatory network, intricate regulatory network that governs biofilm formation. But there's a limitation to what we know, which is that almost all of our detailed knowledge comes from only one Candida albicans strain and its marked derivatives. And so in my lab, we started to wonder whether the same genes are required for biofilm and hypha formation in all strains. And we didn't know the answer. And whether the regulatory network that governs biofilm and hyphal formation is uniform or variable among strains. We didn't know the answer to that. Now, strain variation is well documented and well studied in Candida albicans, and here's just a selection of phenotypes that have been observed to differ among isolates. Drug sensitivity, of course, but pathogenicity, ability to switch phenotypes, mating ability, colonization ability, filamentation, and biofilm formation. And I would encourage all audience members to think about what phenotypes might be interesting and look at those among isolates as well. So in terms of biofilm formation, the story I'll tell you today focuses mainly on 5 different clinical isolates: SC5314, the type strain that all of us are probably familiar with, and then 4 other isolates from different Candida albicans clades. And you can see here in these side views of biofilms that they differ considerably in their ability to make biofilm. So the studies that paved the way for our work, 2 major studies. It was a report from Val McVias and Jerry Fink's lab, that the development of a CRISPR system that allowed very rapid construction of homozygous mutants in Candida albicans, and that was critical for our work. And then the other enabling study that I want to mention is from Matt Hirakawa and Richard Bennett and Christina Cuomo's labs, in which they provided genome sequences for 21 Candida albicans clinical isolates, including those that we're using in our studies. And so that has been a valuable guide. So, are the same genes required for biofilm and hypha formation in all strains? Here is our biofilms formed by a panel of mutants in SC5314. Under our conditions, you can see that BCR1 and UME6 mutations have little impact on biofilm formation. BRG1 and EFG1 mutations have severe impact. In strain 76067, similar kind of outcome to what we saw with SC5314. In this more modest biofilm former, P57055, any of the mutations causes a severe defect in biofilm formation. In this biofilm former, P87, UME6 mutation has a partial defect, BCR1 is more severe, BRG1 and EFG1 more severe still. And You know, I always reflect on the fact this strain, P75010, may have been a bad choice because it makes such a poor biofilm to begin with that it's hard to imagine that a mutation would make the biofilm worse. But indeed, we quantified biofilm formation a variety of ways in hyphal formation, and all 4 of these mutations make biofilm formation worse. So are the same genes required for biofilm and hyphal formation in all strains? We can see that the answer is no. We can see that some genes like BCR1 and UMI6 have variable impact across strains, and other mutations like EFG1 and BRG1 have relatively strong impact across all strains. So when we see a mutation that has different phenotypes and different genetic backgrounds, that's called a genetic background effect. The idea is we make a mutation in one strain, we get one kind of mutant phenotype, and we make it in another strain, we get a different kind of mutant phenotype, or we might not get any kind of visible defect whatsoever. And people have seen these things and thought about them for a long time. And the idea is that each strain, so how do we explain it? Each strain has a constellation of modifier alleles that can govern the same phenotype as the causal mutation that we're studying, like an EFG1 mutation. And so with different constellations of modifier alleles in each genetic background, the impact of the causal mutation on phenotype can vary considerably. So what we think we see then is that for EFG1 or BRG1 mutations, they have quite strong effects and uniform effects on biofilm formation. Modifier alleles have relatively modest effects in the strains that we examined. Whereas for BCR1 or UMI6 mutations, they have quite variable effects on biofilm formation. So those modifier alleles that we hypothesize exist have a much stronger effect on the process. Is the biofilm and hyphal regulatory network uniform or variable among strains? Well, let's take a look. So here's a diagram that represents the outcome of RNA-seq analysis of the wild type and 4 different mutants, the 4 familiar mutants now, in one strain, in this case SC5314. And let me just explain how to look at the diagram so you understand what you're seeing. So in the center of the circle are 4 little boxes. Those represent the 4 transcription factors in which we made mutants and compared RNA-seq data to the wild type strain. And around the perimeter of the circle are about 6,000 little dots, and each one of those dots represents a gene whose RNA level was measured. So there's about 6,000. And where there's a line connecting a transcription factor gene and a dot, that the expression of the gene on the perimeter was altered by the transcription factor gene mutation, altered significantly. So here's what that network looks like for SC5314. Okay. Here's what it looks like for 76067. Looks pretty different. Here's what it looks like for 57055. Looks different still. Here's what it looks like for P87. Still looks different. And here's what it looks like for P75010. Different still. So is the biofilm hyphal regulatory network uniform or variable among strains? At least for the 5 that we've examined, it looks quite variable. And now we've conducted RNA-seq analysis of lots more of mutants in lots more strains, and it looks like it's quite variable across the board. We're not able to group the strains based on clade or other phenotypic properties into simple subsets. So, So now what we think we see is that regardless of the mutation that we're looking at that affects biofilm formation, if we look at a sort of very fine measure of phenotype, biofilm-related gene expression, what we see is that modifier alleles have very strong effects on the gene expression output, regardless of the transcription factor mutation that we've examined. So, of course, it's interesting to describe natural variation, but we'd also like to be able to use natural variation in a variety of different ways. And one way that we've tried to use it is to use it for gene discovery. So how can we do that? Well, I have good news for you. There's lots of different ways. Lots of people have thought about this and taken very productive approaches, many of which are broadly applicable. And I'll tell you about two of the approaches from my lab. So in this case, we're going to focus specifically on EFG1, which is one of the most famous transcription factors in the Candida albicans literature. And it's required for hyphal formation, biofilm formation, and virulence. And when we compared EFG1 mutants and corresponding wild types by RNA-seq in 5 strains, looked at differentially expressed genes, we had about 520 to 860 differentially expressed genes per strain. And as shown in this Venn diagram, there isn't a huge amount of overlap between any one pair of strains. And we found only 177 genes that whose expression was altered in all 5 strains by an EFG1 mutation. We call those principal EFG1 targets. We found also, each strain had its own sort of signature strain-limited EFG1 targets, those genes whose expression was modified by an EFG1 mutation only in those strains. So what buffers the principal targets from those background effects that we've seen? And what makes strain-limited targets susceptible to background effects? Those are really the 2 questions we've tried to address. So for strain-limited targets, we first thought that perhaps EFG1 binds to different sites in the 5 different strains, and that's why it regulates different subsets of genes. And from ChIP-seq analysis that my postdoc Un-Soo Do carried out in 5 strains, you can see that EFG1 binds to essentially exactly the same places in all 5 strains. So EFG1 binding per se is not responsible for the differences in target gene expression that we've seen. So in order to understand what may be responsible, we compared 2 different strains with very different phenotypes, SC5314, which you're all familiar with and is a great biofilm former, and P75010, which is a poor biofilm former. And we looked at genes that were, or transcription factor genes that were differentially expressed between the 2. We found a number, but I'll mention 3 here, BRG1, TEC1, and WAR1, and their RNA levels were relatively high in SC5314 and relatively low in 75010. So we thought that those expression differences may be responsible for the phenotypic differences that we've seen. In fact, if we increased gene dosage of each of those transcription factor genes just by integrating 2 additional copies into the genome, what we found is that we could improve biofilm formation in the poor biofilm former P75010. And you can see that from these side view images on the slide. But BRG1 and TEC1 were well known to be connected to biofilm formation. WAR1 was not as well known. We looked at that a little more carefully. And we actually looked at the regulation of EFG1 responsive genes in strains that differed in WAR1 levels. And as shown here, here's one such gene, ECM331. In SC5314, an EFG1 mutation causes reduced expression of the gene, so EFG1 is a positive regulator of ECM331 under these conditions. In P75010, an EFG1 mutation causes increased expression of ECM331, so EFG1 is a negative regulator of ECM331 under these conditions. In P75010 with our dosage vector, we really saw no effect on ECM331 gene expression. But if we increase the gene dosage of WAR1 in 75010, EFG1 became a positive regulator of ECM331. In other words, increasing WAR1 gene dosage caused P75010 to behave just like SC5314. So a difference in WAR1 levels can account for the difference in EFG1 responsive gene expression. So the way we look at it, the EFG1 transcription factor controls expression of strain-limited targets, and WAR1 levels modulate those and can give different outcomes depending on the strain. And those modifier alleles that we hypothesize exist probably affect WAR1 expression levels. With regard to principal targets, those are the target genes that are common to EFG1 mutants of all 5 strains. And what we observed was that there were a lot of expected genes in that set, including genes that had been connected to biofilm formation and hyphal formation since the dawn of time, ALS1, ALS3, BRG1, and so forth. But there was an unexpected gene, a transcription factor gene called WAR3, and we wondered what was WAR3 doing there. What we found was that if we increased expression of WAR3 just by about 2-fold over wild type in an EFG1 mutant, that we could actually increase expression of EFG1 dependent genes in an EFG1 mutant. So WAR3 could substitute for EFG1 to some extent in this context. In addition, we observed that a WAR3 mutant is defective in biofilm formation. And these are in vitro experiments. The wild type that produces a biofilm, the WAR3 has reduced magnitude, and a complemented strain has increased magnitude. And David Andy's lab also used this mutant in a rat catheter model and got consistent results, about a 10-fold reduction in WAR3 mutant titer compared to wild type in the catheter. So the way we think about WAR3 is this, that EFG1 controls expression of these principal target genes. There are modifier alleles that we hypothesize exist that might modulate their principal target gene expression levels. But EFG1 activity is reinforced by WAR3. So EFG1 can turn on WAR3, and WAR3 then regulates many of the same principal targets that EFG1 regulates. Now, the reason that these studies, I think, are significant, and they're significant for several reasons, but one is that it expands our view of what WAR1 and WAR3 are doing considerably. So WAR1, as you are probably aware, is the sort of central regulator of white to opaque switching. And it's involved in, it promotes formation of sexual biofilms. And WAR1 and EFG1 are antagonists. They repress one another's expression and prevent one another from working. WAR3 is essentially an antagonist of EFG1 in this context because it's a positive regulator of WAR1 expression. So WAR1 and WAR3 are on one side, and EFG1 has an opposing function. And what our studies show is in the context of biofilm, 37 degrees and RPMI plus serum, that EFG1, WAR1, and WAR3 act in conjunction to promote biofilm formation. WAR1 can shape EFG1 function by modulating its interaction with gene expression targets. And WAR3 acts downstream of EFG1 to reinforce EFG1 activity and overcome effects of genetic modifier alleles. So I'd like to leave you with just a way of thinking about using strain variation for gene discovery. So here's that simple sort of paradigm for understanding background effects, that a causal mutation interacts with modifier alleles to bring about a particular phenotype. And, you know, we could just choose any phenotype that seems interesting that we want to study and choose just about any gene with a defined phenotypic impact in the type strain, one that's been well studied or one that we just discovered yesterday. And then, if we think about it, those modifier alleles and their downstream targets have functions related to the gene we chose above. So this is a way to find additional genes that function in the same biological process or pathway. as we found with WAR1 and WAR3 and EFG1. I don't know if I ever told you I love going to work, and here's why. I get to work with really fantastic people. And the projects that I described today were initiated by Manning Wong when he was a graduate student in the lab at Carnegie Mellon. He's now a postdoc with Hiten Madani. And Carol Wolford, a research scientist in the lab at that time, was a consultant on everything that we did, and an incredible help. And at UGA, the work on WAR3 was carried out by Max Cravener, a graduate student who just graduated. The work on WAR1 and EFG1 ChIP-seq was carried out by Lin-Soo Zhou, a postdoc in the lab, and a terrific colleague. I love to be on Zoom and email as well, and here's why. These are the people I get to collaborate with, so I get to talk with them and communicate with them, including Scott Filler, David Andes, Joel McManus, Fred Lonnie, and Damien Chrysan and their lab members. And so let me thank you for your attention and leave you with a little depiction of what that summarizes my talk, and I'll be happy to take any questions.
Great. Thanks very much, Aaron. Oliver, I'll hand it over to you to start off the questions.
Okay, yeah, thank you very much to both of our speakers. And there are already some questions in the Q&A. Please submit further questions whenever you think of them. And I'll start with questions for Darius. And I'm starting right off with an expert in lung immunology. And there are two questions actually about different host-derived molecules. And the first one would be putrescine, which is produced by tissue-resident T cells via, I guess, ornithine decarboxylase. And the question is, what is the role of this molecule putrescine in allergic bronchopulmonary aspergillosis, Darius? Is it toughy for the start?
Yeah, tricky start for 10. So yeah, I don't know is the answer. So I mean, it's a really good point and maybe something we should look at. It hasn't come up in any of the proteomic analysis that we've done, but we can certainly go back through all that data, and we're following up with transcriptomic work as well and metabolomic work. So yeah, we should certainly look at that. It's a good thought.
Okay, and the other molecule is vitamin D, and there is the link to deficiency in the CF patients.
Yeah, so CF patients and more broadly, TB, this is important for lung immunology in terms of respiratory defense. And of course, also in some ways, vitamin D is related to some of the synthetic pathways for ergosterol. So quite an interesting subject, actually. We haven't looked specifically at vitamin D in our models, but again, it's something we should probably look at. And I guess, understanding the interplay of ergosterol is actually a really interesting question that we were thinking about looking at. It's vitamin D2, isn't it, basically?
Yeah, I guess there's a lot of room for looking at different metabolites in this complex lung environment. Okay, here is perhaps a question that is more easy. And this is, how many isolates did you check per patient, especially in cystic fibrosis? These patients often have a long-standing colonization.
Yeah.
Did you check multiple isolates?
So the work I showed you on the phenotypic microarray data is basically from one student whose name is Renat Al-Jahani, who's been looking cross-sectionally. There's another student in follow-up who is basically looking at longitudinal isolates. And from those patients, I think we've got about 20 different patients with longitudinal isolates where we're essentially repeating the whole genomic phenotypic microarray analysis. So that work is in progress at the moment. We did have one patient within the initial data set where there were recurrent isolates, and that patient was a CF patient. And that isolate group did basically have a salt tolerance phenotype, essentially. So they developed, so we've got some evidence that there is adaptation in the host rather than it being some kind of selection from the environment, depending on the patient's endotype.
So I guess this is in-host evolution, but what about, would there be also different strains at the same time with different?
So yeah, in terms of different strains at the same time, mostly we see clonality actually in those longitudinal studies. In terms of the isolates that you isolate in cystic fibrosis patients at one particular time point, then yeah, usually they are clonal, but you do see that you can get some heterogeneity there in terms of different isolates at the same time. It's not the dominant feature.
Okay, that's interesting because it's completely different in some bacterial pathogens in CF.
Yeah, correct.
Okay, yeah, thank you. This was the start for us. And Joe, I hand over to you.
Great, thanks very much. Aaron, the first question is, to what extent does immunocompromise of the host influence how the different strains will change their gene expression networks? In other words, are some of these more or less dynamic in a host with an effective immune response, but not in a host that's susceptible?
Yeah, that's an excellent question. And how any isolate changes in any feature in an immunocompromised versus immunocompetent host is an area of great interest. And we haven't looked at such things, but I know several other labs have, including Anna Selmecki's and Teresa O'Meara's and Scott Filler's. So I await anxiously the publication of their results and hearing them speak at conferences.
Great. All right, another question, Aaron, is, have you looked at protein levels for some of the regulators? And could some of the modifier alleles stabilize or destabilize the effectors?
Yeah, so I think that's an excellent question. And we have looked at protein levels for some of these. Actually, BCR1, UME6, BRG1, and EFG1. Unsu tagged all of those so we could look at their levels on Western blots. We see, small differences in levels. We haven't seen differences in anything dramatic like phosphorylation that we can detect on, at least that we can detect on an SDS gel. But I think it's an excellent point that these modifier alleles are not necessarily just transcription factor genes, but can affect protein modification, phosphorylation, stability, localization, all of the above.
It's going to be fascinating to figure them all out. All right, maybe we'll have one more question for Aaron and then pitch it back to Oliver. The question is, you look at these genes in the context of biofilm formation, which you quantify using your very lovely golden tiara imaging. To what extent is this driven by the straightforward ability to form hyphae? So
we have looked now at 20 clinical isolates, quantified biofilm formation and hyphal formation in various ways. We've looked at lots of mutants in many of those isolates. And there's really an excellent correlation between formation of hyphae, or the extent of formation of hyphae, and the extent of formation of biofilm. The sort of experience as a harsh teacher kind of moment is that the biofilms that we grow, we've started to realize, are really fairly hypoxic. And we usually measure hyphal formation under pretty well oxygenated conditions, 4 hours after induction with shaking. So we, or perhaps our successors in the field, may need to go back sometime and look at this under exactly the same conditions.
Yes. All right, thanks very much. Over to you, Oliver.
Right. So here is a question about the metabolic heterogeneity that you described for the Aspergillus strains, and the question is: could this be related to different growth rates or germination rates rather than real differences in the, let's say, genomic portfolio of these strains, or do you think that's not the case?
Well, I mean, if it is, then it's conditionally dependent. So we assume that, you know, there is some genetic or epigenetic basis for it, because we see differences. I mean, you know, across, you know, different substrates or stressors, essentially. So I don't think it's purely a function of the sort of, you know, quote unquote normal germination rate in normal media, so to speak, if that's what the question is. Having said that, we do see some interesting phenomena, like, for instance, in cystic fibrosis, all of the strains generally germinate more slowly. Why that is, I don't know, but we know that germination rate can modify immunological responses, and, you know, cryptic infections might be one outcome from a slow-growing strain. So, you know, that's an interesting area that's got immunological traction.
Okay, and then I have another question, and this is about the effect you described of the pathogen load on the different immune reaction you get in the patient. So I found this very interesting, and I was wondering how much of this different immune reaction is really pathogen load and how much is genetic predisposition of the patient, because I'm sure that for allergic reactions there should also be some genetic predisposition.
Yeah, so there are you know, genetic alleles. I mean, the obvious one is, you know, cystic fibrosis, obviously, but there are lots of other alleles that actually mostly maps the innate immunity. So there is certainly a genetic component, but I would argue that there's also likely to be a host component as well, and hopefully we can use our animal models to show that. And we're thinking of even, you know, taking that into humans as well. It might be possible, although that's a more long-term aim.
Okay, great. So I hand over back to Joe, but not without a question that is specifically to you about your beard. And this is, when did you grow the whiskers? It's a question by Stu Levitz.
This has been about a year, and so it started last summer.
Perfect. Go ahead with the scientific part.
Yes, it's in honor of Aaron's biofilm talk. That's why I had it on today. So Aaron, there's two related questions about biofilms. One is about the environmental impact on the regulatory network. Have you tested different environmental conditions with your mutants in the different backgrounds?
Yes, to some extent we have. The truth is, if you look at that lovely paper from the Bennett and Cuomo labs, where they characterized, among other things, biofilm formation by these strains, they many of the strains do a poor job in spider medium, which is used very often to study biofilms. And in fact, we ended up using RPMI plus serum, which is outrageously expensive. Because it induces biofilm formation pretty well in all of the isolates that we've examined. But we have looked, and yes, environmental conditions affect the phenotypes of the various mutants as well, and this is totally expected from you know, decades of work on Saccharomyces and Drosophila.
Great. And the related question is, have you tried testing antifungal drug resistance differences in these biofilms? The deeper biofilms look quite porous, while the shallow biofilms might have the same level of resistance because they are so dense.
Now, that's a really interesting idea, and I'm pleased to report I'm walking over to see David Andes this afternoon. So unless the questioner is David Andes himself, I will report back at a later time what David says about it. But he's an expert at this sort of thing, so I think he would be the person to do it.
Absolutely. Well, I can affirm that the questioner is not David Andes, but it's another luminary from the field. So maybe I can jump in with a question for Darius. I was really fascinated by your findings with histidine and histamine. And just curious about the cell types that might be generating the histamine during infection. And is there any role for antihistamines in treating infection?
Yeah, so that's a really interesting question, Joe. So yeah, I mean, it's primarily mast cells in the lung that are generating histamine, actually. Although apparently many other cells will make it. It's just the mast cells make it in really high concentrations, and those mast cells degranulate in response to IgE ligation, essentially. So those are the cells that we're interested in. In the periphery, it's primarily basophils, so you can actually generate those from bone marrow. So we're basically constructing a basophil-based model at the moment to look at that in further detail. And I guess there are broader questions. I mean, we all use antihistamines for allergy, but I'm not sure anyone specifically looked at them in the context of ABPA. And of course, we know that the pathogen itself doesn't have the histamine receptor per se, but it might have a histamine-like GPCR. So there's really a question, I think, with this phenotype about, is this some metabolomic thing? Is it a cellular toxicity that we're seeing with the growth inhibition, or is it actually some kind of physiological response that's mediated through a signaling pathway? And that's kind of where we're trying to head in terms of working out what's going on here.
Wow. So that the idea that you just suggested, the hypothesis that the pathogen might have a GPCR for histamine, that's a very interesting question. And Elaine Bignell, I think, has quite a few mutants of those GPCRs. And
good point.
Christopher Tate at the MRC laboratory has a distinct interest now in solving structures of receptors from pathogens. He's solved the pheromone receptor structures from Saccharomyces, but he's starting to think about solving structures of GPCRs from pathogens. So if you could link one of those to a host signal, I think that would be a real advance for the field.
I'd love to do that if it works out. So I really appreciate your suggestion there. I'm really glad that I mentioned it, and I'm really appreciative of your helpful insights.
Well, I'll just mention one other thread to plant a seed. Some years ago, Don Shepard wrote a Pearls mini review for PLOS pathogens on mast cells, which I've always thought were fascinating, but I also thought it was kind of a niche in immunology. But that Pearls has gone on to be one of the most downloaded PLOS Pearls of all time. Who knew how much fascination there was with mast cells? And might be time for another PLOS Pearls on mast cells and fungal infections. And
yeah, that sounds like a great idea.
Great. All right, there's a couple more questions for Aaron.
Yeah, go ahead.
Maybe I'll pitch it over to Oliver, and you can tee up these questions.
Yeah, I was just going to also ask a question to Aaron, because I find this fascinating, the diversity between the different isolates of Candida albicans, and I was wondering whether you can make any assumptions if this is a specific feature of Candida albicans, or whether you would basically find this in all Candida species, that we really have this strain specificity.
I can tell you my sort of suspicion, not based on a lot of facts, which is that yes, we'll see this diversity everywhere. And really, these studies were inspired by lovely work from Jerry Fink and Charlie Boone and Leah Cowan, in which they compared the Saccharomyces knockout mutant collection phenotypes in two different strain backgrounds. And they actually found some genes that were essential in one strain background and not in another, and lots of phenotypic divergence. And this is again, similar observations have been made, though not as comprehensively, in other organisms as well. And so I suspect that when you get to gene expression networks, that you'll see a lot of divergence among isolates.
So Aaron, we have another question for you in the Q&A, and it is, what are the prospects for systematically identifying modifiers of expression in Candida? In a model where genetic crosses are easy, eQTL analyses are a possibility. That would be difficult in Candida albicans, but perhaps eGWAS or other ideas.
Yeah, so this is an excellent point, that our hands are a little tied just because of the sort of constraints of the sexual cycle in Candida albicans, and picking out candidate genes. I mean, that's what we sort of did, that not based on function so much, but based on expression levels. I think that this is an approach that one can take, and really nice work from Matt Anderson and Richard Bennett has also pursued this kind of strategy. One can use the existing sexual cycle to generate a panel of strains with different alleles. So at least in theory, some sort of QTL analysis would be possible, but I worry that differences in aneuploidy and so forth could be really confounding. It could be a lot of work to make sense out of it.
Right. All right, I've got a question that's two parts. So it's for both Darius and for Aaron, and it has to do with mycoviruses. And Darius, if I remember right, there's a mycovirus that's an Aspergillus fumigatus, an RNA mycovirus. And I'm curious if that might underlie some of the diversity of isolates and clinical presentation that you see. And that would involve going further than sequencing the genomes.
Yeah, so interesting point, and in essence, we are looking at that at the moment. So actually, I've got a student who's just prepping to do a screen across all the 40 isolates we put through the phenotypic microarray, and that is an area of interest because there's some evidence that some of these mycoviruses alter fungal metabolomics. So it would be really interesting to understand that for our organism. So yeah, that's an active process we're undertaking. So very interesting.
So my colleague John McCusker discovered a novel mycovirus in Saccharomyces recently from the 100 Genome Strain Project. And there was only one isolate that had a high level of that double-strand RNA mycovirus, so high that you could see it on an ethidium bromide stained agarose gel. But he found like 98 of the other isolates had the mycovirus at a low level. You could only detect it with RT-PCR. So it seems like you can have 3 different states of a mycovirus, and that might be interesting to look at as well, because there's an active RNAi pathway in Aspergillus that might be controlling.
Yeah. That'd be really interesting.
So that leads into the second half of this question, which is for Aaron. And if I understand it right, at Montreal, there was some really exciting discoveries presented that Candida albicans clinical isolates often have an active RNAi pathway, but SC5314 has a defective RNAi pathway. And Aaron, do you know, has anybody looked for a mycovirus then in SC5314, and maybe even in some of the RNA-seq data, there might be some reads that don't map to the genome?
Yeah, that's a really interesting idea. We haven't done that sort of thing, but I think that would be a good thing for a smart person to do.
Okay. There are a couple more questions that came in the Q&A, and one of them is, are any of your clinical isolates from infected patients, Aaron, or are any of them microbiome isolates? And these two might be the same, of course.
Yeah, all of the isolates that we're using were originally came from David Saul's group, and they are from infected patients infected various sites. Of the 5 that I mentioned, at least 3 are bloodstream isolates. SC5314, as you may know, was isolated by a dermatologist, and dermatologists these days, I don't think, do much work with invasive infections. But from a discussion with Damien Chrysan, what I discovered is that in days gone by, dermatologists were called in to for diagnosis of invasive infections. So maybe SC5314 really is an invasive isolate.
Oh, interesting. I did not know that about the history of SC5314.
Yeah, there's a nice synopsis on the Candida Genome Database website referring to a paper from the then Aberdeen group.
Great. And then one other question, curiosity about, have there been advances in making better medical devices that would have resistance against Candida albicans biofilm formation?
Honestly, I'm not sure that there have been advances that have sort of made it to patients. There have been lots of strategies related to impregnating surfaces with drugs of various sorts, also modifying surface structure as a way of reducing or abolishing adherence, which amazingly enough works. So, but I don't know that those have made it into patients.
Great.
Perfect. So I think we really addressed all the questions. And then I guess we can close the session, not without thanking both speakers. Really cool talks, really broad spectrum of things and to think about. And now we go into the summer break in August, and the next appointment is basically September 28th with the next MycoTalks. Thank you very much to all of you and to the audience.
Thank you. Thanks for the invite.
Very much, Darius and Aaron.